for another week. So no new papers from our group, as far as I know, has been published. I know our mitochondrial paper hopefully should be accepted soon, that a lot of you people are on. Not much news other than our long COVID case report that Al's leading. There's a couple of them. People are interested in joining that case report. We're going to probably be meeting next week sometime. I forget the date, maybe Monday, but we're continuing to work on that. A couple of long COVID cases that the patients that Al sees, it has some interesting phenotypes or interesting conditions that the patients have agreed for us to take a look and publish a case report on. The people interested in joining, the more the merrier. Okay, so let's dive into the recent COVID news. You probably heard, obviously, who declared COVID emergency over, and everywhere's now supposed to go back to normal, but there's been a few good articles. This one, Steve had pointed out to me and other folks, that although COVID emergency is over, we still know hundreds are still dying because of COVID weekly. And then, this is another kind of a reminder that, over the past 32.5 years, 3 years, 15 million excess deaths worldwide was caused by COVID over 2 years, and then more now. But on that note, there's actually an interesting paper there's a few papers that, I saw come out that might be of interest to folks. But this one came out a couple of days ago, and this Nature article here just kind of summarizes it. But we'll go over it for a little bit. But this is like an extremely large study out of the UK led by Kenneth Bally. And this included the cohort that they included were about a GWAS and a TWAS transcriptomic wide analysis. They did 24,000 people was included. I mean, there's actually around 2,000 authors on this paper too, so it's a pretty massive paper. But it was really interesting because they took with their GWAS and TWAS study that they did, they were able to find, kind of key, we all know that, of course, inflammatory immune functions and pathways are key, targets of COVID or key downstream impact of COVID. But they were able to find some genetic leaks and neat new novel markers and genes that previously might not have been identified before with this massive study and the technique. So what's a couple of things, I'm going to go over some of the figures in a second, but a couple of the main highlights, they said, well, JAK1 we kind of knew was already there, but some druggable targets, which, JAK1 was there, but then these other PDE4A or CLS, this immunometabolism related, and also mitochondria related. I know Doug can't make it today, but he probably would have said mitochondria related and other factors. So this is a figure from the paper. One of the figures they're looking at when the 49, and they looked at over 5 different variants for this study, but over time, because there were out of the 24,000 patients, they panned over 5 different SARS-CoV-2 variants. But this is kind of the first kind of dive into it in the figure where they figured out 49 genetic variants, and you can see the over the genomic position, where the genes are, and the specific things related with hospitalization. So it's more severe cases were the case that they're relating to. And the ones, this is like the figure caption from it, but the ones in bold were some of the ones that were previously reported. But then the ones not in bold, you can see there's a lot of genes not in bold here that they've actually identified, which is pretty neat. Power of doing massive analysis. And some of these I know some people might recognize, some people might not. But 3 of the genes that they found were key is here. That's CLS22A31 and that's SFT, which I've never heard of before, and TKY2. So these 3 I hadn't heard before. So novel to me also. And then they looked at the analysis, looked at the structure and different factors, and they looked at how well these genes are being connected to the proteins could be druggable and targeted. So some of these are actually pretty easily using AlphaFold and different computational techniques. They found potentially these are some candidates that could be targeted with some drugs that are already out there. So they did some analysis looking at that. And then, I'm going through this paper really quickly. This is a really big summary overview of it. But what they found with their analysis that in monocytes, because a lot of this patient samples they collected, from the data they got is from the blood or PBMCs and so on. the data really looks at monocyte related tissue markers, and one of them was PDE4A, which I'm not familiar with until now, but it's related with a lot of inflammatory related factors in myeloid cells and so on. And this figure here in the paper was discussing different proteins, key proteins from their analysis that could be potentially druggable in the concentration they have. These are some of the top hits there. So some of these for the inflammation group that we have in our COVID group, the paper we're working on. I know I'm not sure if Steve's here, but I know when I emailed this paper to him, he said, yeah, there's some genes that overlap with that paper that we know of. There's some mitochondrial genes that they didn't talk about mitochondrial deficiency, but we do see mitochondrial genes if you look through here that you might recognize too, or proteins that are here. This is the protein expression. And one of the key strong associations, let's say, this RAB2A gene is related to like the worst outcome in disease, for example, they were saying when they did the ranking of patient outcome and severity and what genes might be more implicated towards that. And they say in their paper that RAB2A is required for viral replication, so that's interesting to read too. But it's an interesting read, and this came out a couple of days ago, or yesterday, or not a couple of days ago, yeah. And it's a pretty massive study, so people can dive into it. They have lots of information in there. Also, obviously, lots of data now is available for people to mine from this big GWAS study too. I don't know if people have seen this data. If they have comments, let me know. I'll keep going. Here's another paper that I think this came out also a couple of days ago, discussing, again, these are all immune and inflammation related, but this paper is talking about how macrophage activation is related with SARS-CoV-2 response, and spike protein might be related to that. And long story short, basically they did some experiments in zebrafish to look at inflammatory process. So they took the spike protein off the SARS-CoV-2 and then did various experiments, and overall they saw that they could be related to activation and oxidative stress, which is of course related to mitochondrial dysfunction. And then summarizing this paper very in a short manner, but they did a lot of experiments and data in this paper. But some of the things they said, they took the peptide, the SARS-CoV-2 spike protein peptide, injected it into larvae, and basically found that macrophages were activated that way, and was downstream impacts was oxidative stress markers and so on. So that was interesting to read. Interesting again with our mitochondrial paper, talking to further things to enhance that story also. This is a paper from some. Oh, sorry. Go ahead. I want to say something. No, okay. Yeah, feel free to dive in if you jump in if you have questions. So this is a paper from, you might recognize some our Brazilian colleagues here from University of Sao Paulo or Campinas, like Marcelo and Helder, that's been in our group and given some talks about their research. But this is a paper that came out, I think this past week, May 15th, on finding the relationship between hyperglycemia and how that might be impacting COVID-19 and the response to it, and how it might induce, let's say, diabetes. And some interesting, I'll go over a couple of figures, or two figures from this paper. But one thing definitely they found, from this, they looked at a patient cohort from, I think it was the Brazilian population, and there was a few hundred people, 600 or so, 200 people. And one thing they looked at, was looking at circulating factors and related with liver function and what could happen with glucose production. And one thing they said, independent of your history of diabetes or beta cell function, this was going to happen. So there was no difference if you had diabetes before you had COVID or SARS-CoV-2 infection, or you didn't before. So this is a mechanism that's regardless of your premorbidities. And basically, they had then looked at one of the concepts that people have been talking about. After your period of time of infection happens, is the virus still replicating in organs? And Rob has shown some places maybe, some places not. Other people have just shown it's the spike part of the SARS-CoV-2 protein is there, but fragments are there and might not be replicating. But what they actually did is find liver from post-mortem patients who died from COVID-19. They died well after the obviously died because of the COVID of the infection of the SARS-CoV-2, but it was after the virus had already supposedly, whether negative, let's put it that way. But in the livers itself, they actually were able to take the virus out of the liver and plaque it out and find out it indeed is replicating from the hepatocytes. So that was interesting. So here's the figure, a figure from that paper. I'm talking about that. So this top panel is talking about if you had died or had diabetic history or were diabetic or not before, or non-diabetic patients, and you see that there's no difference in that pattern for the red, meaning COVID-19 positive or negative, and the impact they have for hyperglycemia. And there's some data here again talking about different glucose levels and other levels related with glycemia and so on. And you can see the different like glucagon and other factors, and you can see this is COVID-19 negative, and these two panels are COVID-19 positive with HG should be high glucose, or NGs or glycemia or non-glycemia. I think that's what it stands for. And you can see there's not much difference there between the factors. And then the other panels are showing detection of viral particles in the liver, and then they have other data, which I'm not showing the figures for that, where they actually then plaque out the virus and they show indeed, it's replicating. So this is probably, I think, some of the pretty good conclusive first evidence I've seen that at least short term after the virus leaves the system, in the livers, the virus is replicating pretty well. And sorry, go ahead. Yeah.
Yeah. With respect to the comorbidities, etc., did they check in this paper for fasting insulin or HOMA-IR, homeostatic model of insulin resistance?
Yeah, it doesn't make a difference. So yeah, I know you harp on that constantly, but it doesn't make a difference here. So again, the premorbid.
Well, I asked.
Yeah. So yeah, I mean, it's been shown that yes, obviously, if you have some premorbidities, COVID-19, we know that already. It's been proven that. But in this case, they're showing hyperglycemia induced by COVID. It doesn't matter about your premorbidities.
Okay, thanks.
So this, we know now XBB.1.15 is, the dominant variant that's happening now. But then, of course, we know the other XBBs that are popping up, 1.16 and 1.9. This paper is just talking about the .1, so it includes. I don't think they had a .116 or the .191 that is up now with this paper when they did the work. But what's interesting is that talking about XBB variants and different factors, they looked at how potential mutations and where these mutations in the virus itself can outsmart or kind of cause the breakthrough through immunity from the previous compared to the previous Omicrons from BA.4, 5, and .2. So this paper goes through different analysis and experiments looking at, for example, this is figure 2 from the paper, looking at some breakthrough infections with the seraphim patients and how this XBB.1 has a pretty good advantage due to the mutations over the previous variants, as we already know. I think they claimed, where they said, but I forgot how many fold increase they had over the previous ones that they talked about in the paper, and these figures show that. And this is the panel that shows the mutations that this XBB.1 has versus other ones, and you can see the phylogenetic tree, the differences, how different it is from the previous versions. But if you're interested in this, I'm sure, I don't know, Nydia, you've seen this or you have comments on this since.
Yeah, I actually just sent something in the chat regarding XBB that the World Health Organization now recommends new COVID vaccines should target only XBB variants. So, yeah.
Oh, I haven't seen that. Oh,
yeah, I just sent the links.
Okay, thanks. Yeah, I mean, it makes sense based on what they're saying in this paper and the breakthrough it has versus because what was it? The last vaccine is BA.4 and 5, right?
Exactly. Yeah.
Yeah, and 2. Sorry. Yeah. So yeah, based on what they're talking about here and what you're saying, yeah, makes sense. I haven't heard of any development of XBB vaccine, so we'll see. I guess maybe in 6 months they'll have a new one.
Yeah,
Who
I
knows?
think this statement was it's just from yesterday. So I think it will take maybe for the winter season, we'll get a
Right,
vaccine, an updated vaccine. Yeah.
right. Oh yeah, and that's, just looking at the fold differences right here in the first line.
Hmm.
It's
Right.
30-fold resistance against the breakthrough, BA.2. Yeah. Yeah, so that makes sense. Although all the reports that have come in, the severity and the outcome of the infection is not too much different than what we've seen from previous ones, other than the getting the pink eye, for example, or conjunctivitis. I
Right.
don't know if you've heard of anything else related to that.
No, just really symptomatology, but nothing on severity.
Right.
Yeah.
So even though it has this 30-fold breakthrough resistance, it doesn't seem like it's causing any more, severity or deaths, I guess you could say, or illness. Long COVID, I
Well,
don't know. But I guess it's time to tell if these new variants, if it does cause long COVID or not, more
right.
or less.
And despite the immune escape, like most people have been exposed to so many variants at this point that it makes sense that it's not as severe. Yeah,
Right,
long
right.
COVID, of course, you never know. Uh,
Although there has been a decline in long COVID cases now, looking at like it's starting to reach a plateau from what we've been seeing. Whether
right.
that's, I don't know why. We don't know why, but yeah.
And also, if I remember correctly, I think in the beginning of the pandemic, people thought that multiple infections would increase your chance of getting long COVID, but
Mhm.
now it's actually the other way around, right? That
Yeah,
there's not such a correlation. So yeah,
yeah. There was a paper, what, a month or two ago talking
yeah,
about that? Yeah. I think I presented it here. Yeah. Yeah, so let's see. Although there's some people still believe that they're holding out. They still believe multiple infections can increase your chance. So
right.
until something
It's also
definitively shows it, yeah, I don't know.
mhm. It's also
Yes, sir.
difficult, like, to assess that since the number of cases has decreased so much. Like, what's the extent of long COVID cases
Right.
from these very few infections?
Exactly. Yeah. So talk about long COVID. Here's I guess some positive news. This is what I just saw this article this morning about Australia is doing their first long COVID diagnostic test launches across the country, so they're testing it out. I was trying to find the paper, and it's based on this paper that had come out almost a year and a half ago now. So it's this blood test supposedly they're doing, and it's looking at these markers here in the monocytes, so the CD14, 16 positive, and S1. I guess this is one of the figures in the paper. So they show that if you have, this is like I think this was the control negative, and this is the severe long COVID acute, but not long COVID, but acute COVID, severe infection. And this, I think, this long bar here is a long COVID of the past, post-acute sequela COVID. So this one here, they're saying and there's more data than this in the paper, but since I guess they based this whole marker on this on this data that came out of this paper, but they have more data showing supposedly if you have this kind of profile and it's up in long COVID patients in the monocytes, I believe you have monocytes, then that's their marker that they're trying to look at. I don't. I guess time will tell how well they could define this as long COVID, but I'm not sure if these markers here will impact other diseases or not. So in this paper, I don't think they'd compare it to, let's say, if someone had the flu or other diseases and how these markers might be elevated. But I'm sure they've done these tests that there's a year and a half later and doing, but maybe they haven't. I don't know if people are familiar with these markers and are more knowledgeable about immunology than I am, since I'm not as knowledgeable as some people may be.
I'm not, but I have a question.
Yeah.
Were these markers obtained before the infection or after the infection?
I think it was. You mean, did they look at it before the infection?
Yeah.
Oh, I don't know. I didn't read the paper too in depth here, but I don't think so. I think they
Yes.
had it. Well, they have COVID negative patients, so I guess that would be this group here, this first group. I think.
Okay.
Yeah.
Right.
But if it's not this plot, but they did look at COVID negative. Oh no, yeah, controls, healthy controls. Yep, HC.
Yeah, it
They
would be
looked at
nice, like, to predict, to
right.
kind of characterize a risk population for long COVID. not
Right.
just after the infection.
Yeah, exactly. So that could, yeah, they only looked at their healthy controls in this paper. I don't know what follow-up studies they've done because I don't see any other publications. So yeah, I guess maybe from this test that they're doing, maybe they'll publish a paper and then maybe they'll answer your question. Maybe I don't know. Or you could reach out to the company. This is Incel DX and ask them. Um, I don't know how willing they are to share data on that, but maybe worth a try.
Right.
But I mean, I guess it's better than nothing, right? But we'll see. Something that I saw interesting. And then also Steve had sent this email to a few of us. If you knew vitamin D is always, it's related to mitochondria, so this might not be a shock. So vitamin D, if it's deficient, there's also an indication of mitochondrial deficiency and things like that, as we know. But this article had come out of this paper. Actually, this paper came out, and these articles talking about this paper saying they did this study that actually going to Twitter. Sometimes people provide good summaries for papers too. And this person had provided a nice graphical summary of this that wasn't in the paper. But they looked at, I forget the patient cohort population they had, but it was a good size. 50 long COVID, there's this 50 non-long COVID, so it's okay size, I should say, not good size. But they basically looked at low vitamin, they found from this patient cohort when they did the analysis that there was low vitamin D levels associated with the majority of, what they say, 50 to 70% of long COVID survivors lack this. So they're recommending, I guess taking vitamin D supplements is not very harmful for the body, obviously. So they're recommending that potentially this could be for long COVID patients at this age group. Could be ways that maybe to people get over long COVID quicker, or at least control the symptoms. That would happen, which makes sense if you say long COVID might be a mitochondrial related disease.
And also, a cofactor, vitamin K2 is strongly supportive of vitamin D3 absorption and function.
Yeah, they didn't find that in this study, but something else that people could look into. Yeah.
Yeah.
So on that related note, there was another study that came out in April, which I missed, but I saw this article that came out like a week ago, talking about, again, it's a group from Cornell Medicine where they using the Recover data, or the Recover initiative, I should say. But this group here, using patient electronic health records available from different areas, and they looked at one from the Florida Plus cohort. They basically, and then compared to I think other regions, they basically saw that depending on your long COVID, basically your long COVID symptoms can vary between, which might not be surprising, between different populations, even around the U.S. So there's some couple of this one plot here just shows different organs and different symptoms that might be incorporated in that depending on the region you are. And this plot here I think is probably what is the finale of it, where they stratified based on sequela over different subgroups, and you can see like male, female, black, white, the overall outpatient, inpatient, and so on. And they're basically in this plot they show that indeed, depending on what you're looking at and what this is just talking about your different health conditions. This is the patient sample diversity, but you can see different sample stratifications and different symptom stratifications depending on what category you want to look at and what different kind of symptom you want to look at. So you can probably stare at this for a little bit and find different patterns. For example, inpatient, outpatient. If you had long COVID dyspnea, it seems like it was more targeting inpatient population, so severe cases. I mean, the majority of these seem as it might be expected. There's more severe cases, as we know, get more long COVID. But joint pain, for example, here is related more of outpatient. So people, but anyway, so you probably stare at this plot here for a little bit and probably download the paper and figure out, see what's happening between different populations, between male, female, white, black, and so on. But it's an interesting paper to read because they use machine learning techniques, as a lot of you people do here, also looking at electronic health records to stratify this and do this analysis. But if you didn't see this paper, it seems interesting to look at the different symptoms. So those are the papers that I saw the past couple of weeks that might be of interest to the group. But then, feel free to jump in if you have questions or thoughts or comments. But we could always wrap up, go circle back at that at the end too. But looking at the variant distribution again, as I said, XBB.1.5, it's the prominent one. It's like a regional map of different places in the U.S. But as we'll see in the following plots coming up, 1.16 and 1.9, 1.1 variants are coming up. The good news is, at least in the U.S., and I think the rest of the world's following suit also, as we know, overall, nationally, this is from the wastewater, which I think is the best indication of what's going to happen now, since the surveillance is very poorly monitored these days, and maybe not as a good of an indication of what's the spikes level to come. But you see overall, it's still a steady decline. It's sort of plateaued a little bit, but going to levels that are definitely pre-Omicron levels are going down. And, trends down. If you look at where I live in Middlesex County area, you see these spikes habits going back up again, but you see the little spikes happen. Maybe next week we go back down, but you see these little bumps that go up and down, but we'll see what happens there. But also on BioBot, they're also now stratifying between XBB.1.16 and .1.9, and then XBB meaning everything else. But it's prominently .1.15. But you can see how XBB.1.9 and 1.16 are creeping up. Still, XBB.1.5 is the dominant one. And this, of course, coincides with what's on the CDC tracker. You can see XBB.1.5 is now dropped to about 70 or 60 to 70% of the variants that are circulating. And you can see the other two variants are creeping up with 1.16, as we saw in India. It's starting to be about 20% maximum, depending on where you're looking at what region, but it's pretty uniform. So that would be probably the next variant, I would imagine, might be dominant in the next few weeks or months. And then this is the last slide. I'm not showing you individual trends anymore, maybe because I think just the global one is pretty telling, and the individual countries are fitting along with this. But you can see, again, new cases. This is not as reliable anymore. Looking at this time frame from last April till now. But you can see, for example, South Korea, there's a little trend going up in cases, but everything else is low. But a lot of the stuff, again, is not as reliable, since everywhere in the world this hasn't been reporting as accurately. So maybe hospital admissions and ICU for the countries that report them is more reliable, or deaths. Deaths, of course, have been low for a while due to all the nice therapeutics and vaccines that people are taking. But you can see, though, the trend for hospitalizations and ICUs are down. That's it for this week.